this video will cover the Ante and Postnatal Screening part of Unit 2, specifically on autosomal recessive inheritance genetic crosses. So better to start off with National 5 revision, because without that you won't be able to pass on that same knowledge into the autosomal recessive genetic crosses and the other ones for later on. So as a bit of a review for this, I've got a pedigree chart, which you probably would call the family tree in National 5, and it's shown inheritance of eye colour, controlled by controlled by one gene, and then represented by two different alleles. So brown eyes is represented by a capital B because brown eyes is dominant, and green eyes is represented by a small b, a lowercase b, because green eyes is recessive. So in this example I've put the squares as males and the circles as females because that'll be the same all the way through all the higher crosses as well. So in this example, and the same with all examples, recessive ones, you will know definitely what their genotype is. And remember, the genotype is which alleles they have and which genes they have. So every person that has a black circle or black cross would then be green-eyed, and therefore their genotype would be two small b's. Okay, as a reminder, their phenotype then would be green eyes, so phenotype is their physical appearance. Every other box or circle, which is the white boxes or circles, would have brown eyes, and therefore would have to have one capital B at very least. Because without that capital dominant B, then they wouldn't have brown eyes, they would have to have green. So from this we'd work our way backwards and look at the children that they have to determine whether they were heterozygous, which is your two different alleles, or whether they're homozygous dominant, which is two of the same allele. So in this, we've got a small b here and a small b here. That means that one parent each must have given the small b to that child. And that small b allele, so that green-eyed allele. So therefore this parent would have to be heterozygous, and so would this parent. Now to determine this child's genotype, we would have to look at what the parents could potentially give. So this parent could either give a large b or a small b. And the same with this parent over here. So this child then could either be homozygous dominant, or it could be heterozygous. Okay, so using the same logic, we can look at these children and determine what these parents had. So because there's two homozygous recessive kids, you can look and see that they would have a recessive allele passed from each parent. So this parent would have to be heterozygous, and so would this one. Then the same way they figured out this one before, if you've got two heterozygous parents, you don't know whether they've each given a capital B, or whether one has given a capital for brown eyes and one has given a recessive for green. So this would be two capital B's, or it'd be heterozygous, so one lower and one uppercase. So as a reminder of National 5 as well, this would be your P generation, the parents to begin with. This would be your F1, and then this second generation, this grandchildren essentially would be F2. As a reminder from National 5 as well, if you were asked to determine or identify people from the F1, this person does not count as an F1 member because they are not related to the parents by blood. Okay, they've married into that, or they're related in by having children. So you wouldn't count these as an F1 member, they would be separate. Okay, so that's your National 5 revision. So now you've got to apply this to your higher genetic crosses. So we'll specifically focus on the autosomal recessive ones for this video, and then we'll do the dominant next time. So autosomal recessive essentially means exactly the same thing as what the National 5 monohybrid cross meant. So there's a few features of this that you'll need to know. First of all, what autosomal means. So this was covered in the last video about diagnostic testing and screening. So autosomes are all chromosomes except the sex chromosomes. So except that X and Y. So in a normal human, you'll have 22 pairs of autosomes and then one pair of sex chromosomes. So autosomal inheritance person would only be affected when it's recessive if they have 2 recessive alleles. Okay, and examples of diseases that are passed in this type of inheritance are things like cystic fibrosis, which is a really common example in questions, and also PKU, which is one we talked about in terms of postnatal screening previously. A typical example question would be talking about 2 unaffected parents and an affected child. So if you have 2 unaffected parents and an affected child, it would always be autosomal recessive. And what that question would normally ask then is, what is the chances of the next child having the condition? So I've given an example of a pedigree chart for this. So we're looking to determine the risk of perhaps other kids having it. So in this example, you would need to be able to identify the genotypes for all of the family members. And the same as before with National 5, this is your P generation. This would be your F1, and again, disregarding the ones that are married in. So this one, for example. And then this would be your F2. So just to kind of break it down, if you look at the genotypes of each person and what they would be, would determine which phenotype. I've given letters that would use here. Okay, circles are females and then squares are males, but for this example that won't matter. So if a person has cystic fibrosis, they would have 2 small f's, and that's because this condition is recessive. If they had a capital F and a small f, they'd be unaffected, and they'd also be unaffected if they had 2 capital F's, so we're homozygous dominant. So each of those would be unaffected. So a person can only have cystic fibrosis if they have 2 recessive alleles, if they are homozygous recessive. So using the key, you'll know that the black circles and squares are people that have cystic fibrosis. So you would know their genotype would be 2 small f's. Okay, and then every other person in that cross would then have a capital at very least, because then that would make them normal. So we'll add the dominant allele to them all, which would give them a normal phenotype and unaffected phenotype. And then you use the rest of the kids or the family members to work out what they are, what the remaining of them would be. So for example, this child here has 2 small f's. They must have inherited one from this parent here, so the other one must have come from the other parent, so therefore this parent here is heterozygous. Same logic applies for this one. There are 2 small f's here. That means that one must have come from one parent up here, and the same for this over here. Okay, same logic with this child. So you can work out all those from there. And the remainder are quite tricky. So for these, for example, if you've got 2 heterozygous parents, you don't know whether they gave each a capital, each a dominant allele, or whether one gave a dominant and one gave a recessive. So your answer here would either be homozygous dominant or heterozygous. And the same applies to this one. So you've got a heterozygous child here. So one parent must have a small f somewhere, but we don't know which one it is. So this one could either be that or this, and the same for this parent. So it would either be homozygous dominant or heterozygous, and the same for this child here, because we don't know what the parents have given to it. Okay, same logic applies over here. Each parent could have given a dominant allele to it and made it homozygous dominant. Or you could have had a parent given one dominant allele and one parent given a recessive allele to make them heterozygous. Okay, now often in a question you might give an example saying that this parent and this parent decided to have another child. What would be the chances of this child being cystic fibrosis affected? So you would do a Punnett Square to calculate this. I'm going to put one parent on the top with their genotypes here, and then the other parent on the side. So if you do the Punnett Square like you would for a National 5, what you'll see is that 1 out of potential of 4 possible genotypes would be cystic fibrosis affected. So therefore, if this parent here and this parent here had another child, it's 25% chance of having cystic fibrosis. So what I'm going to do is stop this one and then move on and do the autosomal dominant next.